Mishra sir. He is the director of ICAR-Directorate of Weed Research, Jabalpur. He served as scientist and senior scientist at ICAR-Directorate of Weed Research, Jabalpur, and then principal scientist during 2008 to 14. After that, he joined as principal scientist at Indian Institute of Millet Research, and after that as head division of crop research at ICAR Research Complex for Eastern Region, Patna. He has many awards and recognition in his credit, like P.S. Deshmukh Young Agronomist Award by Indian Society of Agronomy, then Best Scientist Award by Directorate of Weed Research, Jabalpur, ICFA Distinguished Scientist Award, Editor-in-Chief in Indian Journal of Weed Science, Editor-in-Chief in Current Advances in Agricultural Sciences during 2015 to 17, then Editor of Indian Journal of Agronomy during 2013 to 21. He is the Fellow of Indian Society of Weed Science, Indian Society of Agronomy, Society for Upliftment of Rural Economy, and many more. So his research areas are mainly weed management and conservation agriculture. So with this brief biodata of our speaker, I invite Mishra sir for his lecture on management of parasitic weeds. Sir, please.
Yeah. Okay. Now it is said.
Yes, sir.
Okay, sir. Okay, sir. Okay. Good afternoon, and thank you very much for inviting me again for this lecture. So, after listening, Dr. Das, the president of the society, very comprehensive lecture. Of course, my talk would be on the very specific management of parasitic weeds, and I will try to cover it within, say, 40-45 minutes. So, friends, we all know that parasitic weeds are a different type of plants compared to other weeds, normal weeds, which we can say in the field crops, seasonal weeds. So parasitic weeds are the plants which are depends on the other plants for their sustenance. So they take the nutrients and food materials, water from the host plant. Unlike other weeds where they take the nutrient, water from the soil, here the parasitic plants, they take all these resources from the host plant. So without contributing to the benefit of the host. Because sometimes we take something from somebody and we try to give something to them. So that is not a parasitism. But here, these plants take all the nutrient and water from the host, but they don't give anything to the host, rather than they cause extreme damage to the host plant. So depending upon their nature of parasitism, we broadly classify these weeds into two types. One are the stem parasites and other are root parasites, depending upon their nature of parasitism. So for example, Loranthus, we all know that many of the fruit trees, timber trees are affected by Loranthus. So this is a stem parasite, but semi-parasite because of its leaf green, because of the presence of chlorophyll, it takes most of the photosynthesis, means it manages itself, but takes water and nutrients from the host plant. And in the same way, Striga also being a chlorophyll containing plant, it also partly manages its photosynthesis, but again, nutrient and water and other photosynthesis from the host plant. The other category is root parasite. Oh, of course, sorry, Stryga is a root one. Stryga is a root one, but it is a semi-parasite. The other stem parasite is Cuscuta. It is a hollow parasite because it's completely dependent on the host, because it doesn't have any mechanism, any chlorophyll to produce its photosynthesis. Among root parasite is Stryga, I told. The other root parasite is Orobanki. It also depends upon the host plant completely, unlike Stryga, which partially depend. So broadly, these are the 4 type of parasites. 2 are stem parasite and 2 are root parasite. So just I will deal one by one. Coming first on the Cuscuta, because I have some work on the Cuscuta. So as I said that Cuscuta, also known as Amar Bhel, Akash Bhel, but known by so many names in different regions. So it is a parasite belonging to the family Convolvulaceae or Cuscutaceae. Because of the large number of species, now this family has been recognized as a Cuscutaceae. So the genus Cuscuta, it has more than 175 species worldwide. But in India, there are so many species, maybe 12 species or more. But the most common is Cuscuta campestris. Whatever Cuscuta we see on the field crops, especially Niger or pulses, oil seeds, sisma. So this all belong to Cuscuta campestris. And those which we observe on the small trees or shrubs, they are the Cuscuta reflexa. So these are the 2 prominent Cuscuta species in India. So if we botanically classify how they have been classified, so just take the Cuscuta campestris. So it has 2 styles. All these are botanical terms. Those who are specialists in botany, they can understand more. So 2 style, and the styles are capitate with knobs. Here you can say some knobs, I don't know. Then these flowers, 2 to 3 millimeter in compact heads, 10 to 12 meter across. Then corolla, these are the corolla, they are lobe shape. Then capsule is also round, 2 to 3 millimeter, but they are not connected with the corolla. Then infrastaminal scale, exerted. So all these are some botanical characteristics. In fact, these are required when we want to identify the species of Cuscuta, because these species are identified based on the botanical or floral morphology. So this is the Cuscuta seeds. Here you may be seen it is a bigger seed and bigger fruits, because it is through better microscope, it has been zoomed out. So you can see the Cuscuta fruits. Just here you can see the fruits like amla fruits, but it is not like that. So it comes in bunches, and each bunch has so many seeds. Here you can see bigger seeds, but they are actually very small seeds. And this is another which I was talking about, the Cuscuta species on the shrubs and trees. So this is known as Cuscuta reflexa. So the slight distinction between these 2 species, these Cuscuta reflexa threads are slightly thicker compared to Cuscuta campestris, and also some greenish in color, because Cuscuta campestris is completely pale in color. So this is the most common species found on the woody plants and shrubs, and they are slightly thicker with slight tinge of green color. And you can see the fruits of Cuscuta reflexa. You can clearly differentiate if you remember the earlier slides, because here even flower structure is different, style and stamina are different. You can see the fruit type is also different, and each fruit has only 2 seeds. So they are bigger size fruits and bigger size of seeds. The other biological aspect by which these parasitic weeds pose a difficult task in the management because of their seed dormancy. Not only in Kaskuta, in Orobanki, in Straiga, the seeds are remain dormant for more than 15 to 20 years. So that is why the difficulty in the management. So unlike root parasite, Kaskuta seeds do not require a specific stimulant to induce germination. Even if host is not there, Kaskuta seeds are able to germinate, even without a host plant. A high percentage, often more than 95%, newly matured Kaskuta is impervious to water because very hard seed coat is there. So that's why the seeds which are current season seed, because of this seed coat dormancy, the current season seeds often they may not germinate immediately because of this hard seed coat. Then therefore, if you want to study in laboratory this germination behavior and other things, so therefore the sulfuric acid treatment is required just to making soft their seed coat. So half an hour sulfuric acid treatment may result in 75% seed germination. And we have also seen in our experiment that if Kaskuta seeds are present on the soil surface, then maximum germination is achieved. If it is buried deeper in the soil, the germination may not take place. Here you can see in the photo, this is our own experiment photograph. Even without host plant, Kaskuta seeds are able to germinate and they produce a thread-like seedlings. You can see this is the yellow orange leafless, just thread-like seedlings. And soon after its emergence, the seedling straightens and they try to search the host plant. And if there is no host plant within, say, 8 to 10 days, then they will die. So here you can see, after germination, they try to search the host plant, and when host plant is there, they will try to attach and they will later on detach from the soil surface. So on the contact of the stem, the Kaskuta shoots bind tightly, making 3 complete coils. So just like they completely coils around the host plant stem. And within 2 to 3 days of coiling, then this Astoria begins just to take the required nutrients and water from the host plant. So enzymes from the Kaskuta, this attachment creates some enzyme, so it makes the surface of the host plant soften so that the penetration of Astoria will be easier and it will penetrate to vascular cells of the parasite so that it can extract the food material and water material from the xylem and phloem of the host plant. So under favorable growing situations, many stems may grow from the, you can see here, after attachment, so many stems are coming out from the single point. And once a coil of Astoria, a coil of Kaskuta has made a successful Astorial connection, then the new shoots start, as I told. As Kaskuta grows, it maintains its support by continuously reacting to the host plant, because once it grows, its soil connectivity is checked and then it completely depends upon the host plant. Here just as close up, just I want to show you that, you see, after the attachment of the host plant, this is a view of close Astoria, how this Astoria develops. So this is the onion leaf I took. So here you can see Astoria development in the Kaskuta and then how it is penetrating in the host leaf. And you can see just I pulled out the Kaskuta and you can easily see the teeth, it penetrated in the host plant and it had make a hole in the onion leaf. So because onion is a tender leaf, so just everything is clear here. But the similar mechanism is there wherever Kaskuta attaches the host plant. So in summarized way, this is the germination, then the coiling and the attachment to the host plant. So even the reproductive biology, we study the Cuscuta campestris. So what we have studied in our own experiment, even a single plant produced around 3,696 fruit branches. This is a single branch, this one like this. And even a single bunch has a 17 fruits in a single bunch. And if you calculate the total number of fruits in a single plant, it may be nearly 4,000. And in a single fruit, there may be around 3 seeds. So likewise, the total number of seeds per plant we have calculated is around more than 1 lakh. And this is the 1,000 seed weight, around 4.78 gram. Then Cuscuta may not attach all the plants. There are certain host plants. So like in field crops, all legumes, all your pulse crops like soybean, your chickpea. Of course, in field pea, it doesn't survive. But your chickpea, lentil, soybean, green gram, black gram. Similarly, in oil seeds, Niger is very much preferred host. Of course, ismam, linseed, soybean. And in vegetables, we have seen in onion, brinjal, tomato, lucerne, berseem. Even some of the beets, they also act as a host plant for this Cuscuta. Like you can see the parthenium, you can see the amaranthus, even this Convolvulus arvensis is heavily infested with Cuscuta, and some of the crops like pigeon pea, lucerne, Niger, and some of the hedges. This is our own office building hedge, completely infested with the Cuscuta. So we have tried to estimate the yield losses by the Cuscuta, and we have seen that in chickpea, in field pea, of course in field pea you can see, even in pea some of the varieties, but in French bean there was no loss, even mustard, wheat, so they are not host specific. But you can see the chickpea, lentil, more than 80% loss is there. Similarly, in rice, of course, it is non-host. Cowpea is also non-host. But in other crops, you can see even up to green gram, even in Niger up to 80%. So these are the host plants. We try to find out the extent of losses in the host plant by the Cuscuta. You can see here the infestation of Cuscuta in Niger. Complete dying of the Niger plant. And when you apply the herbicide, you can see how nicely this pendimethalin has controlled the Cuscuta in Niger. So these are the other crops like pigeon pea, chickpea, lentil, lucerne, onion. These are some of the linseed, brinjal, and this convolvulus beet. So we tried to find out the density, Cuscuta density versus losses. And here you can see even a single plant of Cuscuta can cause a sufficient loss around 28%, 39%, even up to 50% in some of the crops. So you can see how dangerous this weed is. So these are some photographs, 1 bead per square meter, 2 bead, 3 bead. So even if 2 bead is there, you are completely, crop like Niger is going to complete loss. So you talk about the management option. So one agronomic management could be, as the Cuscuta can survive without host plant up to 8 days during rainy season. Of course, in summer season, it may not survive more than 4 days because of the dry season. So any crop, say for example, if Cuscuta is there, it is already emerged because it emerges without the host plant. So maybe after 8 to 10 days, if we can plow down it or by stale seedbed technique, so at least those Cuscuta seeds which have already emerged, so that can be managed. Then there was no germination of Cuscuta if it is placed 8 centimeter deeper. Because of the smaller seed, it cannot germinate beyond 8 centimeter. Even most of the germination was there from the surface or just 1 to 2 centimeter. So deep plowing may also be one of the option if it is in your newly infested field. Otherwise, if the field is infested since many years, so maybe the deep plowing may result by bringing a deep layer seed to the surface one. So this is also depending upon the situation, you can use this. Then, of course, this is one of the best technique agronomic management, crop diversification, because we know Cuscuta doesn't parasitize the members of the Poaceae, say like rice, wheat, sorghum, pearl millet. So here crop rotation can be a good technique for management of Cuscuta. So we have to rotate the host crop with non-host crop. And some of the varieties, of course, we tried at our experiment many varieties of chickpea, of course 10 varieties, but none of the varieties of Niger and chickpea showed tolerance. But in linseed, we could try it out of 14 varieties. Some of the varieties were comparatively tolerant. We cannot say it is completely tolerant, but some of the varieties, because of their mechanism, maybe biological, biochemical mechanism, or their hardening of seed coat, or any characteristic of those varieties. So they are partially tolerant to Cuscuta. So this can be used as one of the options in the management. So this is what we did, and you can see here the germination of the Cuscuta. Maximum germination was there, surface, even up to 4 centimeter, it was there. But when it is placed below the 4 centimeter, so you can see germination after 8 centimeter, there was no germination. This is, I was talking about the varieties. So you can see the very susceptible variety like J23 of the linseed, but very tolerant variety like Sweta or the Padmini. So these type of varieties are there which can be used as a part of agronomic management for Cuscuta. Selective herbicides, of course, we know that pendimethalin is there as a pre-emergence, maybe in pulses and oilseeds. It is very good herbicide. So it can be used as a pre-emergence. But if your infestation is there in patches, so there we can use the non-selective herbicide like glyphosate and paraquat. Of course, it will kill the host plant also. But since this infestation is there in patches, so we can use this type of herbicide. But if it is in crop, highly infested, so we have to go for the pre-emergence herbicide like pendimethalin. Maybe in oilseeds, maybe in pulses. So there we can use. Even in Barsim and Lucerne, pendimethalin is very effective, but here the caution is we should use it after 12 to 14 days. Because if we use here as a pre-emergence, it is going to be highly phytotoxic to the Barsim and Lucerne. So not like the pulses and other oilseeds. Here we have to use pendimethalin as a early post means you can say 12 to 14 days after sowing. Then there is one bio compound developed by one of the firm of the Maharashtra Nagpur based. So they have developed a Amarbel, a bio compound, they say. So this is very effective. Even in our lawn, we used this. You can see this picture before application, and this is after application. So it has a very good efficacy, but only caution is it should be applied in the rain-free period, because after its application, if rain comes, the efficacy will be reduced. So it can be applied as a 7 ml per liter of water. So it has been found selective. Similarly, you can see this croton leaves. It was before spray and it was after spray. So this bioproduct is also very effective. And of course, this is one of the other view of the pendimethalin applied in the Niger field. Similarly, in the chickpea, you can see the efficacy of pendimethalin. And here you can see the efficacy of pendimethalin when applied as a pre-emergence. So it is very phytotoxic to the Lucerne and Barsim. But when it is applied as a 14 days after sowing, it has given a fair control of Cuscuta compared to the BD plant. So the same picture which I showed you earlier, this Amarbel bioproduct. So this was all about the Cuscuta in a very brief. So other parasitic weed is a Orobanki. This is a root parasite, complete root parasite. So just I will discuss in brief. So this is a complete holoparasite because it lacks chlorophyll. And there are more than 150 species globally of the Orobanki. But in India, Orobanki ramosa and Orobanki egyptia, they are more common. Then it also prefers the host plant like dicotyledonous plant, especially the plants of the family Fabaceae, Solanaceae, Asteraceae. And in India, this weed has been observed in many states, in many field crops like tobacco in Karnataka, Andhra Pradesh, Tamil Nadu, and Gujarat. In mustard, part of Gujarat, northern Madhya Pradesh, even northwestern Uttar Pradesh, Agra region, and others, and in Rajasthan and Haryana. So this is you can see the picture of the mustard field, how this Orobanki emerges from the roots. And similarly in vegetable crops like brinjal, tomato, potato, so the Karnataka, Madhya Pradesh, and many more areas, this weed is infesting these vegetable crops. It is an annual weed, no doubt, seasonal weed, and it is reproduced by seeds. Seeds are very dark brown and very minute. Of course, Kaskuta seeds are slightly bigger compared to this. This is a like of carbon particles you can see here in the photographs. Millions of seeds are produced by a single inflorescence, and there can remain more than 20 years in the soil. High seed production, as I said, 5 lakh seeds per plant or even more. So radicle emergence can occur within 96 hours. So if the favorable condition is there, its radicle will emerge in the presence of host or a suitable root exudes. Because until and unless a host is there, it secretes root exudates, then only it will germinate, not like the Kaskuta where Kaskuta germinates without host. Here host is required. Even a very brief exposure of the broom rape seed to the host stimulates its seed germination. So after radical emergence, it is 2 to 3 millimeter. Then after emergence, it attaches the host roots. And then again, same story repeats. It will elongate. And then it will develop hastoria. Then hastoria will again attach the roots, penetrate the roots. And it will start drawing the nutrients and water from the root. So here you can see, say this is a flower, then the small seeds. It will come down on the soil surface. Then until and unless these host plant roots secretes, some secretion will be there, then it will trigger the germination. And then after germination, this radicle will emerge and this will attaches the host roots. And again, this host roots will again develop hastoria and it. So around 40-45 days, all these processes will be there inside the soil. When this striga emerges or Orobanki emerges, already at least 40-45 days competition has already been there. So a farmer or an individual, we can see the Orobanki plant only after 45-50 days after sowing. So there are huge yield losses depending upon its infestation, around 30% in tomato, in broad bean 50%, even in mustard and tobacco, depending upon it. Say, for example, in mustard, if it is a purely rain-fed mustard, there will be a huge loss. But if it is a irrigated mustard with optimum dosage of nitrogenous fertilizer, so in spite of the presence of the Orobanki, mustard growth will be more and it is not going to affect more. But in case of rain-fed, the yield loss will be more. So here you can see, even this is, you can see somewhere the host plant may be there. So it has secreted some secretion and then roots are here only. You can see here in tomato very clearly. Tomato plant is here, but Orobanki is here. So it means tomato roots has been reached up to here. So similarly, this is the picture of mustard field in the Rajasthan we collected. So here you can see this Orobanki plant, and this is a mustard root has come here, then it attaches. And you can see how it emerges, because in Rajasthan, Bharatpur areas, light soils are there, sandy soils are there. So they emerges like this, make a smaller cracks, and they will emerge. And similarly, you can see in a single stem of the mustard, a huge branches of Orobanki is there. So why it is so difficult to control? Because, as I said, the farmers' awareness of the problem arises when the parasite is emerging. And we know that it emerges after 45 to 50 days. And by that time, it has done already a lot of damage to the crop. Then huge seed production potential, very small seed. They can move from one place to other place by air, water, and they remain in the soil for more than 20 years. Then the peculiar thing is seed germination is triggered by root exudates from the host and some non-host plant. So these exudates are probably active for a shorter distance from the root surface. So once they are released in the soil, then only the broom rape seeds will be active and they start germination. So even if we thought about the soil treatment, so here you can see that because of broom rape can is stressed even from a 30 centimeter depth. So you have to treat a large volume of soil. If at all you want to consider some of the chemicals that soil can be treated, so large volume of soil has to be treated. And then removal of broom rape by mechanical means is very difficult, because it emerges at the time when already crop is tall one and very difficult to enter in the crop. And in heavy infestation, hand pulling, it is not in fact practical, because already it has caused the tremendous loss to the host plant, and neither it is economical. So this is one technique, but again, soil solarization. But for a larger area, this is not cost effective. But otherwise, it is a very effective technique, because it increases the soil temperature by 8 to 10 degrees centigrade. And by this increase in temperature, the Orobanki seeds become dead because of the high temperature. Then, of course, there are certain trap crops. So these trap crops are grown to reduce the soil seed bank. So these are some of the trap crops like pearl millet, chili, castor. So many trap crops are there. So these can be used to encourage the seed germination of Orobanki. Then trap crop produce the root, because they releases root exudes, and they can cause the suicidal effect of the broom rape, because but cannot find attachment to the roots, because these trap crop stimulate the germination, but these Orobanki cannot attach to these trap crops. So in the absence of host, the suicidal death will be there. Then of course crop rotation is one option, like other parasitic beets, a host with non-host, but at least 2, 3, 4 years, because of the long life seed dormancy. So 1 year, 2 year is not a viable option. Then of course there are some successes here and there to alternating time period, and of course there are some research on that if we allow water flooding for a certain period, then there will be less emergence. And of course the high nitrogen. As I said, in the mustard, we have seen in irrigated mustard, in spite of heavy infestation of Kaskuta, because mustard growth is so high, so the Kaskuta, this Orobanki detrimental effect was not that much. So this nutrient management is one option. And of course, so far we have not came across with the tolerant cultivars for this orobanki. Unlike Kaskuta, we got some success in linseed, but here in broom rape or orobanki. So this is the area where there is a need to work. Of course, the Indian Institute of Rapeseed Mustard, Bharatpur, they are doing some work on developing tolerant cultivars on the broom rape, against the broom rape. So here some of the very old slides, work has been done in some other countries. So here you can see the soil temperature increase. So because of the soil rise, at least 10 degree, more than even 10 degree temperature increased. And because of this increase in temperature, you can see the broom rape shoots emergence was almost zero. It was zero compared to non-solarized. And this was the yield. So you can see where the non-solarized plant, non-solarized situation was there. It was full of orobanki and there was no yield. But in case of solarized, because it completely controlled the orobanki, so there was yield. So this is one of the good techniques, but again I was telling you, it can be used for a smaller areas, for a vegetable small areas or high value crops. This technique will work well. Otherwise, for a larger area, it may not be a cost effective. So here you can see the manual pulling. It is a farmer of Junjunu district of Rajasthan. You see how beautiful this mustard crop is there. But if you enter inside, these type of orobanki are there, but they may cause some loss. Of course, they may create huge seed production for the next crop. Soil fumigation may be one option, but as I said, a huge soil volume has to be taken into consideration. So that may not be a practical aspect. Then some studies in other countries use of seed germination stimulants like ethylene, estrigol, gibberellic acid. But again, how to treat a large mass of soil by these stimulants? So this is also a big question mark. So some of these herbicides like trifluorine, fluchlorine, or pendimethalin, they have been found effective, but in a varying degree of effectiveness. Somewhere people reported they are partially effective, some were good, but some are not at all effective. So depending upon the situation. Similarly, our Haryana Center, HAU Center has done a lot of work on the lower dosage of glyphosate in mustard. So they, of course, this is a reference from other countries, but even in mustard, Haryana people has done and they got a good success. But again, question comes because glyphosate is a non-selective herbicide. It is not registered to be used in the mustard field. So for research, it's done. It's okay, it can work, but for recommendation point of view, we cannot recommend it. So these are some of the examples of the trap crops against different species of orobanki. And these are some of the catch crops that can be taken in between the two crops. So this was all about in nutshell, about the orobanki management. And in the same way, just I will discuss in very brief about this striga. Because the mode of attachment, mode of interference and damage is almost same with the orobanki. Because both are the root parasite. The only difference is orobanki is complete root parasite and this striga is a partial root parasite. So here you can see, see the picture of sugarcane. This I took from the Hyderabad, one district, Nizamabad district. You can see in the heavy infestation of striga, it is how devastating the sugarcane crop. Similarly, at Modipuram Institute, in direct seed upland rice, I don't know from where this wheat came in the rice, but this photograph I took from there. So in rice field also, this Striga infestation was found. Although Striga is mainly from the hard sub-Saharan countries, Africa, in parts of Asia. Earlier it was even in semi-arid areas like parts of the Tamil Nadu, Andhra Pradesh, Telangana, where this maize, sorghum, pearl millet, sugar cane, and some of the upland rice, it was reported. Of course, species may vary. For example, in African countries, this Striga hermonthia is a major issue because there is sorghum and pearl millet, these are major crops and almost dry land crops, rain-fed crops. And if it is not managed, 100% crop failure has been reported. Some of these studies in other countries, they have estimated the economic loss. Of course, very old loss. If you further upgrade it, it will be many more millions. So 50 million hectares in 300 million farmers are affected in Striga in Africa. So they have estimated the loss more than $7 billion US. So similarly in other crops also. In India, of course, a very old study. Then because if you see earlier, sorghum, particularly in Rabi sorghum, Striga infestation was more, especially in parts of Maharashtra, Ahmednagar part and other parts, and of course in Telangana. But nowadays, in sorghum, because of the hybrid sorghum and others, its population is drastically reduced. So just like I discussed with the Orobanki, same is the case here. So after maturity, these seeds will be there. They are the dormant seed. Then they require a conditioning. So stimulants will be produced by these roots, and then they will start germination. This radicle will come up. You can see. Then it will come in contact with the host root, and then it will attach it, and then it starts Astoria formation. This Astoria will penetrate and will penetrate up to the xylem of the root. And then it will start sucking up your nutrients and water and other metabolites from the host plant. And this is the whole life cycle, how it develops. So when we talk about management, hand weeding, very difficult. Then use of trap crops here also, like groundnut, desmodium, cover crops, then intercropping. These are some of the studies with the host crop like maize and sorghum. So they have been found to reduce its infestation. Similarly, trap cropping of cotton. So there have been reports that 90% of Striga seed bank has been reduced by trap cropping. Then if it is low fertility or resource poor, means resource-less soil, so low fertility, low moisture, so there Striga infestation will be more. Then again, like Orobanki, if you apply more nutrients, more nitrogen, so there its growth will be reduced because simply we can say the host plant growth will be more, so it will have detrimental effect on the parasite. Then of course in Striga we can go, because Striga affect mostly on the plants of the Poaceae family. So there because it is a broadleaf bead, we consider it. So here we can apply 2,4-D as a post-emergence. But only point is, when it emerges from the soil, maybe 45-50 days after, so by that time it has already caused a lot of damage to the host. But by this way you can reduce the seed bank for the future generation. Then in some countries there are studies that if we do seed coating of imezothapyr resistant maize with imezothapyr, because in some of the countries there has been advanced research where they have developed the imezothapyr resistant maize. So there they can go for imezothapyr coating of seeds. So this will be a very effective strategy for the, say for example, just I am saying, for example, we in India we have developed a imezothapyr tolerant rice. whether by the IRI or NRI Cuttack or Savannah seed industry. So there, if we feel that in some of the area, rice growing area, this striga is going to be a problem. So there we can try whether imagetapire seed coating to that rice seeds is going to reduce the striga problem. So that is a point of research which we can do. Similarly, a seed treatment with imidazoline herbicide. So it is again in some other countries where this type of research work has been done. So these herbicides can be used for the management of striga, especially in maize. This is another view of the striga infestation in sugarcane. In Indian condition, as agronomic management, we can go for intercropping of sugarcane with cowpea, groundnut, soybean, because these are the trap crops which allow striga to germinate, but they are not going to parasitize. So these crops can help to reduce the seed bank in the soil. Then, of course, incorporation of organic manures and other things also, heavy dose of fertilizers. So they will boost the crop and they will provide the competitive ability in favor of the crop and they will suppress the weeds. Similarly, the high dose of nitrogen, 2,4-D, we have already discussed. Our one of the ICRP center at US Dharwad. So they have developed a microbial consortia, particularly striga. And they have observed, they have tested that even if this consortia at the rate of 20 kilogram per hectare can be used with 500 kg of compost at the time of sugarcane planting. So it has a very good effect on the emergence of this striga. And of course, some of these studies at our TNAU center has been done, and they have come up with the pre-emergence of, in sugarcane, in fact, pre-emergence application of atrazine plus 1 beating, followed by earthing up, then combined with post-emergence application of 2,4-D, along with the urea at 90 days after planting. So this is very effective strategy. And similarly, mixed application of atrazine plus 2,4-D or metribuzin plus 2,4-D at 100 days after transplant, after planting of the sugarcane. So this is, they have been found very effective against the striga management in sugarcane. So these are this. So this is one of the technology we say, push and pull technology, of course, intercropping of desmodium with maize. Some of the other countries' technology is there. So here they intercrop desmodium with maize. And they, on the bunds, they plant this Napier or Bracharia. So by this way, they manage the insect. Because once insect will sit on this Napier or Bracharia, they cannot, means their egg laying and other pupation process will be adversely affected. So they cannot become the major pest of this. And desmodium also releases the chemicals into the soil that stops striga. So it means these intercropping, they release some chemicals so that are detrimental for the striga. So these are some of the techniques which we can also try in India, because this desmodium produces again a defense mechanism. So whatever insect are about to come on the maize, so because of this defense order, they will be repelled. And if they want to sit on this border, they will not be able to hatch their egg laying and another hatching process. So by this way, the complete management of striga and insect will be there. Then the last one, just I will discuss the Loranthus. This is also becoming a major pest, major parasitic weed in plantation crops, in fruit trees, in timber trees, in coastal side. If you go to the sites of this Odisha from Konark to Bhubaneswar, or if you visit the Goa and other parts, a lot of timber trees are affected by the this Loranthus, and there are so many species in Loranthus. Even in our villages, Earlier day, we remember that out of so many mango trees, hardly 1% or 2% trees will be affected. But now, after maybe 20, 30 years later, hardly 1 or 2% trees are left with the orobanking. Otherwise, almost all trees are infested with the this Loranthus. So this situation is very severe in the trees. So there are so many species in the Loranthus. In India, more than 70 species. You can see different type of species are there from different trees. And attacking several hundreds of trees and extending from the sea level, as I discussed, in the coastal area to as high as Himalayan region. So among these, Dendropthy pelcata, this is one species, sorry, which is more common in fruits, ornamental and forest trees. And this plant, in fact, it bears very beautiful flower. And of course, this again the biology, having this much of long length, so on and so forth. So the parasite, it starts flowering from June to December. So depending upon the host range. So maybe in mango, it may flower maybe June, July, or in some other trees in different times. It's again, seeds is red in color. So this is mostly liked by birds. So its movement, seed movement from one tree to other tree is mostly by the birds. So the bird sits on the trees. So the seeds in the bark of the tree, because of the moisture, then seed starts germinate. So you can see the infestation of this Loranthus in mango tree. This is the Indian Institute of Sugarcane Lucknow research. So I took this photograph on the mango. So how devastating it for the mango. So almost all the forest trees, high value timbers like teak, rose, sandalwood, eucalyptus, casuarina, neem. So you name any tree, they all are the preferred host for this beet. Similarly, in fruit trees like mango, citrus, sapota, pomegranate. So every tree now you will find this infestation of this. So depending upon their presence, it will affect the foliage, phenology, reduce growth, yield, quality. Even in timber, this make a knot and reduces the timber quality. So wood quality is also affected by producing large knots, and it will alter the physical properties. Mango cashew plantation, if you see, I was talking about this Konark to Bhubaneswar, whole, if you visit the coastal side, all cashew plantation is there. And I think most of the cashew trees are affected by this beet. These are also one of the slides I took from I was earlier in Patna. So my fruit center is there at Ranchi. So here you can see the mango tree affected by this Loranthus. And here we see that this Loranthus acted as a host for this black fly. So on the back side of the leaf of Loranthus, these are the eggs of the black fly. So this is one of the black fly is one of the insect which causes the tremendous loss to the mango fruits and others. So apart from the direct loss, it also acts as a host. So not like the field crops, we can recommend so many herbicides like that, and we can go for directory spray. So because it affects the timber trees or fruit trees stem, so a lot of work has been done by our Bengaluru center. So there they have suggested is carve the bark of the parasite at the point of attachment, where it is attachment and astoria is there. So there you have to scrape the bark. And you have to go for padding with cotton containing 4 gram of copper sulfate plus 0.5 gram of 240 sodium salt. So you can have to, you can do this padding. So slowly and slowly, your this Loranthus will die. Then we have to directly spraying of ethereal wherever possible. Because what difficulty is in these fruit trees, to go to the site of attachment, it is very difficult to climb on the tree. And most of the cases we have seen if you see the tree branches, at the last tip of the branches, this parasite will be there. So it is very difficult to go there and go for petting and other all those things. Then the final solution will be lopping off the affected portion of the tree in the initial stage. So therefore, the monitoring, we have to do the strict monitoring. And as soon as we see that the particular branch of a particular tree is affected, the best way is to remove that particular branch. I think this is one of the best solution for management of this weed. So by summing up, I can just say that the parasitic beets are more difficult to control, because as I said in beginning that these are not the beets of other seasonal beets of seasonal crops. So these are the parasite because they have the connection with the host by way of their physical connection by twining, then biochemical, biophysical, and of course there may be some genetic connection also with the host plant. And under changing climate, when we talk about the temperature is going to increase, carbon dioxide going to increase. So there we expect that because of the high temperature, as we know that the Striga and Orobanki, they prefer the hot and arid climate. So maybe under changing climate scenario, the problem of Striga and Orobanki will be more, and they may move from the those areas where warmer and hot climate will be there. Then, of course, the conventional weed control strategies like our cultural, chemical, agronomic manipulation. So they are not that much effective. Of course, the crop rotation, intercropping, trap crop, soil solarization, green manuring, they can give some relief, but they cannot be ultimate solution of this crop. So therefore, with the advancement of the technology, now the training is our smart weed management. So in some of the countries, or even in India, some of the research on the biotechnology aspect is going on. So their better understanding underlying the chemical, molecular, and other factors that control pre and post attachment interaction. Because certainly we know that when host and parasitic plant attachment is there, so there must be some interaction. So therefore, before going for any study, we have to understand properly what type of interaction is there, how they are affecting each other, how they are penetrating the hostoria in the stem or roots. So that is very much required. Then with the advancement in genomics, so this will allow a comprehensive understanding of the molecular biology of the host plant interaction. Then with the genomics study, with advancement, so we have a lot of literature available on gene silencing techniques. So maybe through double standard RNA. So maybe biotechnologists people may know better because I'm a agronomist, so I cannot understand much of these technologies. But these are some of the hope of the ray, or the ray of hopes, where such technologies can be inculcated in the host plant or parasitic plant, because ultimately, if anything is happening, certain genes are responsible for the penetration or host parasitic relationship. Can we silence those genes by any mechanism? Because then those relationships can be break. So this type of research may be going on in some other parts of the world. So that type of study are needed. And similarly, the host parasitic signaling genes and genes shared by host and parasitic beads, they have been identified by some of the workers. So how these identified genes can be integrated or can be inculcated in the host plant or parasitic plant to develop a parasitic bead resistant biotype or variety. So I think these are some of the smart and future perspective to develop a integrated approach or to develop a smart approach for management of parasitic beads. And wonder this is the world largest flower, that is the reflexia. Reflexia, you can see how beautiful this flower is. And this is a parasitic plant. So thank you very much for your patience hearing. So if any comment, any other things are there, I will be there to answer. Thank you very much.
Thank you very much, sir, for giving very comprehensive lecture on parasitic weed like Coscuta, Orobanchis striga lorenthus. A very in-depth knowledge was given in this lecture. However, there are challenges also for the management of these weeds and possibilities also through advanced approaches. So thank you very much for giving insight into this topic. So on behalf of all the participants and organizers, I would like to thank our speaker, Dr. J.S. Mishra sir, for giving very informative lecture. Thank you very much, sir.
You may take some questions, if
okay. If participants they have any questions, so they can raise their hand.
Even they can put in the chat box also.
Yeah.
So
yes, sir. So we have 4 participants. So Miss Kavita Sagar, you can ask your question.
So thank you so much. It was rather indeed very interesting topic, especially the parasitic weeds. And as per your conclusion, there are many challenges in management of parasitic weeds. So rather this was very interesting and newer topic for me as a weed a personality individual who is working in the field of weed. So my main one common question is, you discussed that Cuscuta species, it damages the host cells through enzymes, releasing enzymes. Which could be those enzymes? Are you talking about the cell wall damage, which could be pectinase or some other enzymes, sir?
No, actually, those enzymes help the Astoria to penetrate the parasite into the host, because these parasites once attaches the host plant, so they secrete some of the enzymes so that their host plant peripheral cells, maybe the cortex or other things, they may become loose, and that may allow the penetration of the Astoria. And once Astoria is penetrated in the host stem, so they will extract whatever nutrients and water or photosynthesis is there. So they are not directly damaging the host plant enzyme or anything else. Enzyme secreted by the Cuscuta, they felicitate Cuscuta to penetrate its host. It's a Astoria to the host to draw the nutrients and water from the host plant, making the host plant bigger.
Yes, sir. Yes, sir. That's my exact question is, which could be those enzymes which are secreted by Cuscuta in order to...
that exactly, I don't know. In fact, we have to verify it.
Yeah, because when it has to enter into, rupture the host cell, it has to go through first outermost layer of the bark,
Cortex,
maybe the cell. Yeah. Maybe the outer layer of the plant, it will be containing sometimes cutin, sometimes some host will be containing pectin.
Right.
Yeah, probably they would release cutinase, pectinase, cellulase, hemicellulase.
Yeah, that's right. That's
right. Yeah. Yes. Thank you, sir.
Thank you.
Jota Srilata, you can also ask. Mr. Thakkar Singh.
Hello, am I audible?
Yes.
Sir, would you give me some reason why POSC are unaffected by the Kaskuta?
One reason maybe exactly that's what I told in the earlier madam asked the question. Because you see, exactly biochemical aspect I have not studied, but there may be because of the bark structure or stem structure that may not allow to coil the, or even after coiling that may not allow the penetration of the Astoria, or even that may not allow the development of the teeth in the Astoria. So in fact, that biochemical aspect, in fact, I don't know much. So I have to refer the literature or other those biochemist people who have done this, because maybe one physical reason may be because of their hard seed coat or backseal layer or anything that the Astoria may not be penetrating. But what are the chemicals or what are the other biochemical reasons in the upper layer of the cuticle or that exactly, in fact, I have to study. Thank you very much.
Thank you.
Miss Pemula Priyanka, you can ask.
Good evening, sir. Sir, my question is, in Kaskuta management practices, we have studied about stale seed bedding, sir. So I am thinking in Kaskuta, stale seed bedding can be done because it doesn't require any stimulation for germination. But coming to other parasitic weeds, they need stimulation for germination, sir. But I
Yeah.
think stale seed bedding is not possible. Is there any artificial stimulants for germination of these parasitic weeds, sir, other than Kaskuta, for stale seed bedding like that? Any artificial stimulants are there, sir?
Artificial stimulant, just I have studied in the literature because, say, for example, strigal. In one of the slides I shown you, strigal and gibberellic acid, those are there. So by petri dish study or by part study, we can study it. But because we have to treat a large soil volume, because orobanki or striga seeds may be present up to a 30 centimeter or 15 centimeter soil depth. So practically it is not possible to treat that much of soil volume by this strigal or gibberellic acid or any other stimulant. For research purposes, we can do it in the small part experiment others. So those studies have been concluded that strigal, gibberellic acid. So these are some of these stimulant which can stimulate the germination of the orobanki and striga seed. But practical difficulty is how to practice it in a large volume of soil.
Okay, sir. Thank you, sir.
Mr. Nikhilesh. Mr. Nikhilesh, you can ask. Or Miss Pemula Priyanka. Okay, sir. If they are unable to ask. So, sir, we will close.
Even if somebody has not asked and if he has some doubt, so he can share those doubts in the chat box so that
Yes. Participants, you can share your questions or doubts in chat box. We'll try to answer those questions. So, sir, now we'll close this session. So, thank you very much, sir.
Thank you.